Welcome, everybody. This is Rotary E-Club of Silicon Valley, and every week we bring you the story of people who are making a difference in the lives of others, making their communities better, and finding wildly cool things to tell us about and inspire us to see the world in new ways, which is very much what we've got going on today. I should introduce us. We, as the Rotary E-Club of Silicon Valley, we're one of 36,000 clubs around the world, 1.4 million Rotarians and Rotaractors who are very much pledged to serving others in all sorts of cool and interesting ways. And speaking of cool and interesting, we're back to our speaker. Our speaker is Dr. Hank Greely, who is a professor at Stanford, both of genetics and law. He is the director of the Center for Law and the Biosciences at Stanford. And there is one line that or one pair of sentences I just have to read on our way into it. You read most of the bio as you were coming to this recording, but from the write-up, humans will be risk-averse about editing ourselves, but much less constrained about editing other species. We will rewrite the DNA of the world's biosphere for reasons good, bad, and frivolous, and our current regulatory system is nowhere near prepared to handle this. Nothing but cool and interesting stuff on the way. Hank, thank you so much for joining us. We hand the microphone over to you.
Thank you for inviting me. Over the last 20,000 years, our species has almost completely remade the biosphere of the Earth. Right now, something like 90% of the mass of all mammals living on land is humans and human livestock. 20,000 years ago, it was 1%. We've changed everything by the way we've changed the environment. But now we have new tools that will let us change it even more. And so what I want to talk about today really is 4 things in 12 minutes. I want to tell you a little bit about the genome editing tools that we're going to use to do this, that we're already beginning to use to do this. I want to talk about its use in living humans. I want to talk about its use in future humans. And then I want to talk about its use in the 99.9999% of the life forms on Earth that aren't human. So first, genome editing. Mainly, right now, we're talking about CRISPR, which is a wonderful acronym. It's wonderful because the actual phrase is impossible to remember and talk about and say. The clustered regularly interspaced short palindromic repeats. I'm very grateful that the scientists who named it with an I to a good pronounceable acronym. CRISPR is a fascinating tool, the most important biological tool created in this century, at least. It is a combination of Model T Ford, a Swiss Army knife, and a word processor routine. So it's a Model T Ford in that it is not the first genome editing device we've had. That was arguably invented at Stanford 50 years ago to move genes from one organism to another. And over the 50 years, over the following 40 years, newer and somewhat better ways were adopted, but they all were clunky and expensive and time-consuming. CRISPR, first published in 2012 by Jennifer Doudna at the University of California at Berkeley and Emmanuel Charpentier, then at Umeå University in Sweden, is just better. It's faster, it's cheaper, it's more accurate, it's easier to use. And that's why it's like a Model T Ford. Before the Model T, there were cars. There were cars for 25 years before the Model T, but there weren't very many of them because they were really expensive and they broke down all the time. So not only did you have to be rich to buy one, you needed to be able to afford to have an on-house, 24/7 chauffeur mechanic. Henry Ford comes up with the Model T. Suddenly, everybody can have a car, and pretty soon everybody does have a car. Cars have changed our world enormously. I'm a Californian. Cars have shaped everything about California, including the air we breathe. But it's not the first car that did it. It was the accessibility of cars broadly through the Model T. CRISPR makes genome editing accessible to high school students, to 20,000 laboratories around the world, to potentially bioterrorists, to just about anybody who's willing to learn a little biology and spend a few $1,000s. Second, CRISPR is a Swiss Army knife. CRISPR does lots of different things, and we're discovering new things for CRISPR to do every day. The one I'm talking about, though, is CRISPR for editing DNA, and that's the word processor routine. CRISPR is like Microsoft Word's routines that find, cut, and replace. So one piece of CRISPR finds a specific location in DNA, a specific set of A's, C's, G's, and T's. Maybe it's A, T, T, C, A, G, G, A, C, T, A, G. It finds it and it holds it. And then another piece of CRISPR, a CRISPR-associated protein, cuts it at that point. And then another piece of CRISPR another part of the CRISPR complex can put a new piece of DNA where you took the old one out. So let's say I get a manuscript from somebody to review, and it's misspelled my name. Instead of Greely, it's Greeley. If it's on Word, I can tell Word, find every place it's Greeley, and it will. Then I can tell it, delete every place it's Greeley, and it will. And then I tell it, replace it with Greely, and it will. That's CRISPR's main use. It's not perfect, but it's awfully good. So second, how are we going to use this? Well, on humans, we're already beginning to use it to change people's DNA, mainly to try to treat disease. It is a better way to do something we've been working on for over 40 years called gene therapy. So if someone, let's say, has cystic fibrosis, they've got a particular gene called CFTR that's spelled wrong in a way that means it doesn't work and they get sick and die. With CRISPR, you can go in, take out the bits of the DNA that are spelled wrong and replace them with bits of the DNA that are spelled right, and presumably fix, cure the patient and the disease. We're not there yet with CF, but we're getting there. We're not there yet with sickle cell, but we're getting there. Gene therapy's been tried for 40 years. There are now 3 or 4 FDA-approved gene therapies out there. CRISPR is going to make new gene therapies much, much faster and easier. And so that's the exciting part. There are going to be some pluses and minuses. Blood diseases are relatively easy to treat because you can take blood cells out, fix them in the lab, check them in the lab, and put them back in and have them take over your blood system. Lung cells, you can't take the lungs out and fix them in the lab and put them back in, or at least not easily. So there'll be some things that are easier and some things that are harder, but a lot of the genetic diseases are going to be cured or at least seriously treated thanks to CRISPR. Third, CRISPR in unborn people. This has been the most controversial area, so-called germline genome editing. Now, the germline is basically your eggs and your sperm. It's the only part of you that lives on past you. All the other 35 trillion human cells that you have die when you die. So if I change your blood-forming stem cells to fix your sickle cell disease, that change is nice, it helps you, but when you die of old age at 90 instead of at 19, it dies with you. If I change your sperm or your eggs, that can pass on to any children you have, or grandchildren, or great-grandchildren, or great-great-grandchildren. Doesn't have to. Maybe you won't have the kids. Maybe they'll get different genes and not that one from their parents, but it can pass on. And that's made it a red flag for many people who think that we cannot let ourselves change future generations. This was first done almost exactly 4 years ago, or first announced by a Chinese scientist named He Zhonghui. And those of you who speak Mandarin, please forgive me for butchering his name. That's the closest I can come. Who announced that he had CRISPRed the genomes of 2 embryos that became twin girls. It's 4 years ago. He said they were healthy then, probably lied in part. How are they now? We don't know, because the Chinese government is releasing no information in order to protect their privacy. It was a huge outcry. Interestingly, the first newspaper article in Beijing said Chinese scientist leads the world. The next one, after 122 Chinese academics had posted an open letter on WeChat, their version of Facebook, talking about how this embarrasses our country and shames us, said Chinese scientist suspended, under investigation, and ultimately he was sentenced to 3 years of prison. So some people think this is a terrible thing. Some people think it's a terrible thing now. Nobody thinks it was a good thing for He to have done except He. But we don't know whether it's safe and effective. And the most unethical thing you can do to people is to do things that are unreasonably unsafe or not reasonably effective. So everybody agrees, don't do this now. But some people say never do it because it affects future generations. And some people say, well, if it's proven safe and effective, maybe there are times when it could be used. This is a hot fight. My own view on it gets me shot at from both sides. I don't think affecting future generations is particularly significant because there is no the human germline genome like the Ark of the Covenant. There are 6.8 billion germline genomes. Each one of us has one, and it changes each generation. The genes that I have and pass to my kids are different from the ones that my parents passed to me, because in the 30 years between when I was born and when I started having kids, those sperm went through a lot of mutations and there were changes. We change it and we affect the changes by our culture. By moving to agriculture, humans ate a lot more starch. And as a result, we have 4, 5, 6 copies of a gene that makes a protein that helps us digest starch. Our ancestors 20,000 years ago had 1 or 0. Agriculture changed our genome. Insulin changes our genome by letting people with type 1 diabetes live long enough to have kids who have a higher genetic risk statistically for type 1 diabetes and would never have existed but for medicine. So I don't think it's should be a red line. On the other hand, I don't think it's going to be used very much because almost everything we could do through changing the genes of embryos or of sperm and eggs, we can do through changing the genes of babies through gene therapy, or we could do in other ways. There are other ways we can try to fix these problems. Now, on both of these, living humans and future humans, big question everybody has is, what about enhancement? It's nice you're fixing diseases, but are you going to make super babies? Everybody's interested in super babies, good super babies and bad super babies. The answer is maybe someday, but not in my lifetime. And I don't think in the lifetime of anybody listening to this, anybody who's alive in 2022 and listening to this, because we don't know the genes for super babies. And the things we most care about, behaviors, are really complicated, involve hundreds, if not thousands, of different genetic variations. We don't understand it. We might in 20 years, we may better in 50 years, we may never understand some of the complex stuff like intelligence. So yes, enhancement might be a worry, but it's our grandkids' worry. And I'm content to let them worry about it. Last point, the rest of the biosphere. As Rushton said at the beginning, I think we're going to do a lot more with that because we're not so worried about bad cattle, bad calves, or bad baby turtles, or bad bacteria, or bad mosquitoes. We're really worried about damaged babies. We're really risk averse with babies, and that makes perfect sense. But we're not so scared about having other species have birth defects, so we're willing to risk more. We will do it. We'll do it for good reasons, like agricultural reasons. It'll be a lot easier with climate change to change the genes of corn to make it more heat tolerant than to move the corn belt 300 miles north. It'll be a lot easier to change Cabernet so it can still grow in Napa Valley and they don't have to move to British Columbia with their Cabernet vines. It'll be a lot better in lots of ways. I think this will even lead the Europeans to eat genetically modified food. But we'll also use it for other things. We'll use it to try to get rid of, say, mosquitoes that carry malaria. We'll use it to try to get rid of rats that are eating the native birds of New Zealand. We'll use it to try to make biofuels. We'll use it to make green poodles. Why? Because we're a frivolous species. We can, we will. Somebody will make green poodles. Rose breeders for centuries have been trying to breed true blue roses. Nobody's succeeded. CRISPR should make that easy. We will make woolly mammoths, not soon, but in 20 or 30 years or something, a close approximation of woolly mammoths. We will make dragons. They won't fly. We're not going to repeal the laws of physics, but they'll look like dragons. And some of us will make biological weapons. So there's good, bad, really ugly, and frivolous. And we will do all of it because that's who we are. The problem is we've got a decent regulatory system almost everywhere for messing with humans. We have crummy regulatory systems for messing with non-humans. In the US in particular, we grossly overregulate animals, underregulate plants, and don't regulate the rest of life forms at all. That's got to change. Though how we get a sensible, nuanced bill through Congress, I'm willing to take advice on how that happens. So thanks for your time and attention. Keep an eye on this space. We have changed life enormously. We're about to change it 100 times more enormously. It's not necessarily a good thing or a bad thing. It's going to be some of both, but it's good for us to go into it with our eyes open and some sense, some ability to control what happens. Thank you.
Thank you so much, Hank. Now, I am excited for us to get into the questions. I will introduce the folks that we have on the recording real quick. So from our club, there are three of us. Our secretary, Mike Kane, feel free to wave. The Paella Master, Shag Shagrin, and me, Rushton Hurley, the program's chair. Now, the first question that came in through the chat is one that is about CRISPR. If we assume that there will be bad actors doing less than altruistic applications of this technology, what are possible avenues for regulating this kind of behavior so that we can hopefully head off some of the more frightening horror movie style approaches to this?
So if they're truly bad actors, terrorist groups, individual wacko terrorists with a college biology degree, or terrorist nations, we can try. We won't, there's no guarantee we can succeed. Murder has been illegal for a real long time, but murders still happen. But we're working on it. The best way to use CRISPR involves ordering from some central lab an RNA that is the thing that sticks to the DNA you're looking for, that finds it, the guide that grabs what you want. You order it from a lab and they send it to you. There's a list in the industry of DNA and RNA sequences that the labs, if somebody orders them, they're supposed to call the FBI, because these would be things found in like smallpox, for example. So we've got some cautions along those lines. The FBI is very interested in these possibilities of bioterrorism. Scientists are concerned about it, and I think there's increasing interest in kind of promoting a culture of whistleblowing about deeply unethical work. But I think it's going to be hard. There are literally tens, if not hundreds of thousands of labs in high schools, colleges, and universities and companies around the world that can do CRISPR. And there's not a really good choke point through which to control it. So it'll be difficult. I'm actually probably at least as worried about people with not bad motives who make a mistake, and they unleash by mistake the next kudzu or cane toad into the world. They've changed something and it turns out not to be the good thing they hoped, but something bad. And again, there's no great way to do that. I think we could have a regulatory system that required some sort of licensure, registration, a little bit of protocol review, some testing before you put anything out into the environment. You can keep it in your lab, okay, but if you let it out, you've got to show that it's kind of safe and effective, and maybe most importantly, a plan for what to do when things go wrong. That plan might be something as simple as making sure you've got lots and lots of insurance, but or something so simple as putting RFID chips into the mammoths you release into the wild. But we need to think about it, and right now we're nowhere close to being able to deal with it on the non-human side. The regulatory stuff on the human side is pretty good. Although obviously, as the Chinese example from 4 years ago showed, not perfect, though I will note that China has really upped its game in biotech regulation since then. Whether it'll be effective regulation or not remains to be seen, but they've certainly talked the talk since then.
So you make this distinction between the willingness to augment, with regard to diseases, what we do in kind of human space, and how how much less we may have in terms of constraints for non-humans. With the augmentation that happens in non-humans, what are some of the particular concerns that you would say you have related to what you have read about how people are using CRISPR in their own research?
Okay, so there is a way to use CRISPR with a variant called a gene drive that I won't explain here, but if you Google gene drive, and particularly a guy named Kevin Elster, Elster, you get lots of information about it. It makes it much more effective in passing from generation to generation. People are really excited about using it to wipe out mosquitoes. You could actually make a change that makes all mosquitoes that get born male. So nice thing, male mosquitoes don't bite you, but even more importantly, if there are no female mosquitoes, after a few years, there are no mosquitoes. So that sounds like a good thing, but maybe mosquitoes turn out to be important for ways that we don't recognize, that we don't understand yet. Maybe somehow the species that you wanted to get, Anopheles gambia, which carries malaria, maybe that gene drive somehow moves over into a neighboring species that you didn't want to wipe out. So I worry about letting things out before we know enough about them. And there is research going on with gene drive in various ways. I worry about, I don't worry very much about it in the US because we've got, well, I worry about letting plants out that may have consequences that we don't understand. We had been regulating that fairly well. We still regulate it with respect to plants that have a gene moved from one species to another. But US Department of Agriculture has said it's not paying attention if you just edit genes that are already in there. It's only taking care of ones where you moved it from one species to another, which is the old-fashioned way that we're not going to be doing very much. There's a guy who I think is trying to do good work to cure a genetic disease in dogs. It's a big dog, I forget exactly which one, but there's a known genetic disease. He's trying to fix it. Well, with no regulation, with the current regulation in the US, he can't do it unless he raises $80 million. And it takes 10 years. He's a dog breeder. With no regulation, he might be able to do it, but it might lead to lots of pain and suffering in the dogs, or it might turn out that what you've changed in the dog turns out 10 years later to cause something worse. So it's the unforeseen consequences that I think we need to pay attention to, completely apart from the ones that really scare me, which are, the bioterrorists. And I am scared about good people making mistakes. I'm more scared. I go back and forth as to which scares me most, but bad people trying to do this for bad reasons. Supposedly when ISIS took over Mosul, they took over the med school there, which was the second biggest med school in Iraq. And supposedly they killed the head of the science department there because he refused to help them make biological weapons.
So you mentioned at one point along the way the genetically modified food, and perhaps even the Europeans, might eat this stuff at some point.
Yeah.
But can you tell us more about concerns that may be there related to genetically modified food, and how testing is obviating that?
Sure. First, everything we eat has been genetically modified. Almost all of it by those brilliant geneticists, our ancestors, farmers and herdsmen.
Right.
Wheat is a human-made hybrid of 3 different grasses. Chickens look nothing like their ancestors, even like their ancestors 100 years ago or 80 years ago. We've modified them to be much fatter, plumper, faster to grow, juicier, all those things we like. Even things like fish we catch in the ocean. There's a great story, a great study of a commercially fished species in the Atlantic, and it was losing the number of individuals in the species. It was diminishing badly. So they put in a requirement that if the fish is below a certain size, you have to throw it back. The idea was these would be juveniles, you'd throw them back, they'd make the next generation. Well, that did help some, but it's also turned out that over the years, now the adult fish are smaller and smaller because the adult fish that were small are the ones that got thrown back and bred and had small kids. So genetic modification, even when we don't expect it, we do that to all of our food. I think the two things that are legitimate concerns about genetic modification of food, one is allergens, which are screened for and tested for currently. And the other is bad environmental effects, for which there have also been a variety of tests. It's harder to do tests on the environmental effects because you can't put a whole section of Iowa farmland into a lab. But people have worked on it. I would note that actually we should have been doing that for a long time because breeding changes things in ways that could be dangerous as well. It's not just the genetic, it's not the way you change the genes, it's the fact that you've changed the genes. And hybrid corn has been around for a long time. X-ray modified, X-ray mutations in crops have been around for a long time. We haven't worried about those. The real worries that people seem to have are much less scientifically founded and are based on the idea that it's unnatural, by and large, and that unnatural is bad for you. And of course, everything we eat is unnatural. Everything about us is unnatural. This is not my fur. These are not my eyes. You and I are not talking face to face. What humans seem to be most prone to do is to try to insulate ourselves from nature and make things better for us. So I think there are some concerns, appropriate concerns about GM food. I don't think they're very big. I think they're fairly well dealt with now, but attention needs to be paid to them. But a lot of it is, I think, a less rational, more emotional reaction. And I've got no problem if somebody wants to eat what they think of as an all-natural diet. I just have a problem when they impose that diet on everybody else, which the EU has largely done, not just to the EU, but also to a bunch of their trading partners in Africa, where they say, don't take GM food, or we'll cut off some benefits for you.
Well, let's get one more question in before we finish up the recording. So you've been paying attention to these kinds of technological advances for years. And over that time, I would guess that at points along the way, you have been surprised by what people have managed to accomplish that before you found out what they could do, you really did not have the sense that that was a possibility. Are there particular surprises in the history of the development of CRISPR technology or gene editing in general that stand out for you?
Yeah. First, I'd note that much more frequently, I've been surprised that things that look like they can't miss have missed. So if 1 in 50 things announced as a breakthrough actually turns out to be important, you're doing pretty well. Most things that are ballyhooed as breakthroughs turn out to be of modest interest or none at all. But I'd say the fascinating thing about CRISPR to me is nobody went into CRISPR studies with the idea of coming up with a transformative tool. It was first noticed in the 80s by some Japanese scientists. A yogurt manufacturer, Danone, did some research on it because they were having trouble with some of their yogurt cultures. A microbiologist in Spain named Francisco Mojica found some weird residues and some bacteria he studied. None of this, except maybe the yogurt, had any kind of foreseen application. It turns out CRISPR was invented not by Doudna and Charpentier, but by bacteria 3 billion years ago to help protect themselves against viruses. What happened then is Doudna, who is an RNA microbiologist, microbiochemist, and Charpentier looked at this and said, we could use this as a tool. And so after 30 years of people looking at CRISPR thinking it was just purely of academic interest, it's become an enormous world-changing technology. And I view that as a real vote for continuing to support pure, basic, apparently not particularly useful research. Most of the time, it will turn out not to be very useful, but every once in a while, it'll change the world. And CRISPR is a nice example of that, I think.
Fantastic. So as we finish up, Hank, I'll hand it back to you at the very end for a final word, just whatever you'd like for people to kind of have in their heads as they leave the recording. But for all of you who have been watching this recording, thank you for joining us. We hope that you are intrigued by the thoughts that you have and that have been shared, and that you will share them with us. There is a discussion forum at the bottom of the page on the Rotary, SiliconValleyRotary.com website, where you are perhaps seeing this meeting. And if so, let us know. Let us know what you think about this or any other element of the meeting. Also, let us know that you attended. There's an attendance form, and Rotarians, if you are looking to make up a miss from your own club by properly typing in your email address, it'll generate an email that you can pass along to your club secretary, and all good there. Hank, we love having great thoughts come and shared with us, and you have certainly done so today. Thank you. How would you like us to finish off the recording?
All of you listening, I hope you'll pay attention to this area in the future. This area being bioscience, life science. We know approximately infinitely more today than we did 30 years ago. 30 years from now, they'll look back and say, those fools in 2022, they thought they understood something. They knew nothing. This is exploding. There are other areas that are exploding, like computer science and information technology. But this one's about us. We are biological organisms. Most of us are not computer programs. And so it really it speaks to us and the knowledge that's going on. There are great science journalists out there who write very accessible stuff. Carl Zimmer, Ed Young, and others, and even some stuff by academics can occasionally be accessible. So read about it, follow it in the newspaper. Not only is it important, but if you catch the bug, as I did 30 years ago, it's really fun. So enjoy.
Wonderful. And everyone, we will see you next week.